Hi, this is Dr. Swapna and I welcome you to your Learn Lab. If you are an IGNOU student looking for help in your studies, then you have reached the right place. Here, we have lecture videos on different core and elective subjects of important IGNOU courses. We aim to help you ace your exams. So hit subscribe to our channel and as you watch, make sure to like and share too. Come and join me on this beautiful learning journey. In this video, we are going to do block 3, which is population and sampling. Within block 3, we have the first unit, which is unit 6 of the same title, population and sampling. Let's see what all we are going to cover in this video. So we'll be seeing what is a population. We'll see what is a sample. We'll also see what is sample size and sample errors and how we determine the sample size and why sample size is important for a research. We'll also see what is sample bias and how it appears and how to avoid them. We'll also see what are experimental and control groups. Let's start with the first topic, which is what is population. So population refers to the collection of all elements under a study having one or more common characteristics. For example, you are conducting a study on the stress level of students in a school. This is an hypothetical situation. In this situation, all the students who are studying in that school becomes your population. Or maybe you are doing a study on all the people living in a particular part of a state or maybe a geographical region. In such cases, all the people who you find in that particular geographical area comes under your population. So the population here is what is the literal meaning of it, how you know how you define a population of a country or a state. That way, same way, in a research, any element or all the units that come under the ambit of your study is the population, and total number or elements in a population is known as the population size. All right, and there is no limit to the size of population. It can be anything. There is no limit there. Moving forward, so population in research is defined as a well-defined collection of objects or individuals with similar characteristics. For example, you are studying the effect of drug on anxiety, in which case your population will be every individual who is suffering from anxiety. So that would be the similar characteristic or the characteristic based on which you will be identifying your population, which is presence of anxiety. Again, so population in research is defined as a well-defined collection of objects or individuals with similar characteristics. It is a large group of people, objects, or individuals who are the primary focus of a scientific examination. All right. So based on your study, your objective or your purpose, you identify the population which you're going to study. Now let's look at the types of population. So first we have finite population. So as the name suggests, finite population means there would be a fixed number of individuals in the population which you can count. You can determine the population size. The number would be finite. Here in infinite population, you will not be able to find exactly what is the population size or what is the number of individuals or the number of objects which are present in the population. It will be uncountable. Okay, so you cannot determine the population size in this case, in infinite population. In case of finite, you will be able to determine the population size. It would be a fixed number. Again, existent population, which means this is the population that exists, that you can observe, okay, which is not made up or abstract or something, you are imagining. It's there. They exist and anybody can see them or see it. All right. When it comes to hypothetical population, it is a population that does not exist. This is something you are creating out of your imagination for the sake of hypothesizing something. It is a hypothetical, which is actually not there. It's just your imagination or something you are using as a part of your study. Maybe like for example, let's imagine we have a village where everyone is still, I mean, in this century also everybody is involved in barter system and nobody is actually going out and earning money. The village doesn't believe in money. So that would be a hypothetical situation which may not exist in reality, but something that you are imagining or making up for the sake of your study. So that is hypothetical population. Now that we have seen what is a population, let's see what is a sample. So a sample refers to a subgroup or subset of a given population. So when you have a population, that would be a large number of individuals or units which you have to study. In cases where it is not possible for you to conduct study on all those individuals in this population, what you do, you will take a representative sample or a subgroup or a subset. Now this representative subgroup or subset is called a sample. It signifies a part of the population that is chosen at random to participate in a study. Now, this is the most important part of it. The sample must be chosen at random, which means every single individual or every single unit in the population has the equal probability to be a part of the sample. There is no like a person has higher probability or the other person may not be chosen at all. Nothing of that sort. Every single individual will have equal probability. There's an equal chance that anybody can be chosen for the sample or for the study. And generalizations about the population are inferred from the observations made from the sample, which means whatever outcome you get from the sample is supposed to hold true for the population also. Because the sample we have taken is representing the population, whatever study you're conducting and whatever outcome you get, it is, you can generalize it, you can extend it to the whole population. So sample is an important part of a research study where you are not able to perform the study on the whole population because of the large population size. Now, these are the three important characteristics of a sample, that the sample should represent all the characteristics of the population, which means the sample should be a true representation of the population. Whatever characteristics are exhibited by the population should be exhibited completely by the sample also. Otherwise, it will not be a true sample. All right. And it should be free from any bias, which means it should have been chosen at random. So there is no bias of the researcher involved in selecting the sample from the population. The sample size should be sufficient to allow statistical analysis. It should not be too small or it should not be too large also. If it is too large, then there is no point in calling it a sample, right? So the size should be sufficient enough so that it allows efficient statistical analysis. Now, let's see what is sampling. So sampling is the process of choosing a sample from a larger population. No brainer there, right? It's called sampling, which means this is the process of selecting the sample from the larger population. Each individual under observation is known as the sampling unit, which means if you have a sample of 10, and in this each of them would be called as sampling unit. And how you choose these 10 from the larger population is called sampling. The purpose of sampling: it makes research feasible and efficient. This means you are saving your efforts here, because suppose in case you have a population of 10,000 individuals, it would not be possible for you to conduct your interview or maybe experiment on all these 10,000 people. This will make it very tedious and it will not be feasible also for you. And efficiency will also be less in if you have to like go through all these 10,000 people and you alone are observing, I mean making all the observations and performing the whole study. This can reduce the efficiency also. So when you are choosing a sample of 100 from this 10,000 folks, you are making the study feasible and efficient. It also saves cost and time, right? Time definitely, cost of all the resources that you would be using. It allows representative conclusion. This means whatever you are getting the result for these 100, they would be generalized to the total population also. Why? Because this sample is representing this population. They are true sample or the true representation of the population, which exhibits all the characteristics that is there in the population also. It also helps in in-depth understanding, which means when the sample size is good enough for a researcher to handle, the researcher can plan deeper studies, or they can make deeper observations and generalizations also, because the units that are under study is manageable for the researcher. So this is the purpose of sampling, why we do sampling and why it is necessary when your population size is big. Let's look at types of sampling now. So we have two main types of sampling techniques. First is non-probability sampling and the second is probability sampling. Now we'll be studying in detail about this in our next unit, which would be the next video. But here, just for the sake of introduction, I'm going to differentiate between non-probability sampling and probability sampling. So the non-probability sampling is a technique where each member of the population does not have an equal chance to get selected. While here in probability sampling, each member of the population will have an equal chance to get selected. So here's what's happening. In a population of 100 people maybe, each of those 100 people will not have an equal chance or probability to be the part of a study. While here, each and every individual of that 100 will have an equal chance to get selected for the study. Secondly, the sample does not completely represent the target population if we are using non-probability sampling. All right. So here what happens is that the researcher is using his own bias or a selective criteria to choose a sample, in which case it may not truly represent the target population, or it will not show all the characteristics of the population. While here, the sample does completely represent the target population, which means here the sampling is random. Each and every individual has an equal probability to be chosen for the study. So the sample will show every single characteristic of the population. They will be a true representation of the population. Thirdly, the individuals were chosen deliberately by the researcher to take part in the study. Here, the individuals are chosen without bias and completely randomized. So, if I want to perform a study in a university, maybe like on what basis people select courses. So, if so, ideally, what I'm supposed to do is each and every student of the university should have an equal probability to be a part of my study. But I don't want to put all that effort. So what I'm going to do is I'll just choose people who I know from the university and ask them to be a part of my study. So what happens now? I am using a bias or maybe my convenience. So I'm deliberately choosing certain individuals over others, in which case I am using non-probability sampling, right? While if I am going to go for probability sampling, what I'll do is I'll put up a notice and I'll invite everyone to be a part of my study, so that there is no bias here, randomized. Everybody has an equal chance to be a part of my study, right? So that is probability sampling. Then generally, the non-probability sampling is applied for qualitative research. Qualitative research such as what? Case studies, pilot studies, and for developing hypothesis. Alright. While probability sampling is generally applied for both qualitative as well as quantitative research, where statistical analysis is conducted and generalizability is desired. Alright. Why generalizability is desired? Because when you are using probability sampling, the sample would be a true representation of the population, in which case whatever results you are getting for the sample can be extended for the whole population also, right? While here in non-probability sampling, we will be using it for qualitative research, where you do not want to do any statistical analysis and you are not looking for generalizability, just for developing a hypothesis which you will test later. All right. Then non-probability sampling is also used in studies that do not have an interest in the parameters of the total population, which doesn't consider what the total population look like. But here in probability sampling, it is used in studies that consider the parameters of the total population. Why? Again, because we have the sample which is a true representation of the population, where all the parameters of the population will be exhibited by the sample also. Here, the parameters may not be exhibited by the sample. Non-probability sampling is easy, quick, and less costly, right? So I'm just choosing people I know. Just takes a phone call, or maybe when I meet them, I can just say, okay, yeah, please be a part of my study. Here, it is tedious, time consuming, and costly also. All right. So that's the difference between non-probability sampling and probability sampling. We have seen what is a population, what is a population size. We have seen what is a sample, what is sampling, types of sampling, and now let's come to sample size. So because we know population size, it's pretty much easy for you to guess what is sample size also, right? So yeah, this is simply the size of the sample. So it's defined as the count of total individual samples or the number of observation that are used in a survey or experiment. Okay, so it's the count of the total individual samples in a or the number of observation that are used in a survey or experiment. Total number of individuals in a sample. All right, that is sample size. It is always a positive integer and is denoted by the variable n. Why? Because it is a number, right? It will always be positive. You cannot have negative one number. Okay, so it's always a positive integer. No exact sample size as it can vary as per the research setting. Okay, so we cannot generalize that the ideal sample size should be this or that, because the ideal sample size will differ and vary according to the research or study that is being conducted. If all conditions remain the same, if all the conditions remain the same, which means if the sampling is random and true representation, then larger the sample size, higher is the precision in estimating the properties of the target population. Doesn't mean that sample size should be equal to population size, no, but larger the sample, more precise your generalizability or estimation of the population would be. Okay, so yeah, that was the significance of sample size. Going forward, how to determine the sample size? So we determine the sample size based on the circumstances and objectives of the research, right? I told you there is no fixed or ideal sample size. It will vary or differ based on the research. Now, both non-statistical and statistical variables are used for determining the sample size. All right. So the non-statistical variable includes manpower, which means how many people you have got to help you in your research, budget, which would be the money involved, availability of resources. It could be any resource. It could be your stationery, or maybe the travel contingency, or maybe the literature. Any of it could be a resource. Anything that you use would be a resource. And then sampling frame. So sampling frame is the actual list of all accessible units within a population from which the sample is drawn. So in a huge population, what all parts are accessible for you from which you will be drawing the sample makes up the research frame. And this is crucial for ensuring that the selected sample accurately represents the target population, and this enhances the generalizability of research findings. Okay, so lastly, the non-statistical variable would be ethics, and we have studied what is ethics, right? So the research, whatever you're conducting, should be beneficial for the society. It should not harm anyone, right? So yeah, these are the non-statistical variables based on which you will be selecting your sample size. The statistical variables that we use to determine the sample size are desired precision from the estimate of existence. This means the error margin. How much error are we allowing? How much error is acceptable in the outcome? Secondly, the expected existence of the phenomena, like how frequent or what is the expectation that the phenomena exist in the population and the sample. All right. So these two are the statistical variables that we used to determine the sample size. Next, we look at the criteria to infer the appropriate sample size. So the first criteria we have is level of precision. We have already seen this, right? So this is the sampling error, or the estimated range in which the true value of the population lies, and is denoted in percentage points. So this is the estimated range in which the true value of the population lies. All right. So this is the level of precision. Next, we have confidence level or the risk level. This is the statistical measure that signifies results from an action that are supposed to fall inside a pre-mentioned range. Which means if we say our confidence level is 95%, which means out of 100, 95 times our results hold true. There is a 5% chance that there is some error and it is wrong, or there is an error in the outcome. So this is confidence level. It is the statistical measure that signifies results from an action that are supposed to fall inside a pre-mentioned range. So whenever we are going to start a research or study, we first decide on the confidence level. Like how much are we able to take the risk of getting a negative result or a wrong result, right? And finally, we have degree of variability. This means how spread out the data points are within a set or group. Like for example, if this is our what we have 10 values, what is the difference between each of these values? How far they are from each other, or how close each of those points are to each other. This is what is meant by how spread out the data points are within a set or group. All right. Next, let's see what is a sampling error. So by name itself, we know what is a sampling error. It refers to the deviation of the chosen sample from the original traits or characteristics or qualities or behaviors or figures of the total population, which means how different the sample is from the population. Ideally, it should not be, because when you're choosing a sample from the population, you want it to be a true representation. But when there is a sampling error, the sample will not have all the traits, characteristics, or qualities, or behaviors, or figures of the population. And why does sampling error happens? It happens due to sampling bias. And how can sampling error be eliminated? So ideal case, it's very difficult to forego the sampling error, and if you actually want to forego the sampling error, then you have to forego the sampling, which means you have to conduct the study on the entire population and not go for a sample. If you perform the study on entire population, you will prevent sampling error, but there is a chance of other errors coming up. All right. So this is practically not possible in most cases, but it can be minimized. Sampling error can be minimized by employing unbiased probability sampling, which means the researcher is not using any bias or any deliberate features to select the sample from the population. The researcher has to use probability sampling, where each and every individual or unit has an equal probability of being selected for the research. The second thing that can be done is taking a larger sample size. So we have seen larger the sample size, less is the chance of error, more is the generalizability. So sampling error cannot be totally eliminated if you are choosing a sample from the population, but you can minimize it by using random sampling or taking a larger sample size. Then let's look at the sampling criteria. The characteristics based on which the sample is selected is called the sampling criteria. And the criteria is determined by what? The research problem or the purpose of research. So based on the research, it could be either age, right? The sampling criteria, I might want a sample of particular age group. So if I am studying adolescents, then all the adolescents or the age group of adolescents becomes my sampling criteria. If I am doing my study on a particular gender, then that becomes my sampling criteria, right? If I am doing my research on a particular profession, then everybody who is pursuing that particular profession becomes my sampling criteria, right? So based on the objective of the research, the researcher can decide on the sampling criteria or the feature or the characteristics based on which a sample is selected from the population. All right. Next we have sampling bias. So this unit is mostly just small basic topics, right? So there is nothing to go into detail in any of these. So very basic unit, just as an introduction to the coming unit or the unit 7 on sampling techniques, right? So yeah, we'll look at sampling bias now. What is sampling bias? This is the error that occurs when the sample chosen for study is not representative of the larger population it is supposed to represent. Alright, so when the sample that is being chosen is not a true representation of the population that it is supposed to represent, then that is called the sampling bias. In case of an unbiased sample, in case there is no sampling bias, the difference between the individual selected from a sample and the total population they represent will be accidental or by chance. So we said that whenever the sample is selected, it has to be a true representation of the population. In case of a sampling bias, if the sample you are choosing is not a true representation, that is because of the error. But in case you have chosen the sample randomly and there is no sampling bias, in such cases, if there is a difference between an individual, the characteristics of an individual and the characteristics of the population that the person belonged to is purely accidental or random or by chance. Alright. If the reason for the difference is anything other than chance, then there exists a bias in the sample. Alright. So if the difference is not due to random chance, then there is a sampling bias that has happened. The researcher has not gone for a sampling that is probability sampling. Alright. Let's see the causes of the sampling bias. The first one we have already discussed, non-random selection methods, which means we are using methods wherein every individual of the population do not have a fair chance or an equal chance to be a part of the study. Second is convenience sampling, which means the researcher is using his or her own convenience for selecting the sample. So if I am using only people I personally know for my study, that's convenience sampling. If I'm opting for students in my class for my study, not going into other classes or other schools, then that is my convenience also. Right? So convenience is when the researcher is not going out of their comfort zone and want to get all the sample without having to work for it. All right, using their own convenience. Thirdly, using leading questions. So leading questions are, questions which are phrased in such a way that it subtly, it subtly points towards the expected answer from the participants, which means it would be, influencing the participant to provide a specific or a predetermined answer, right? So the participant will not be given a free choice, but subtly, pointing fingers at, okay, this is what you need to say, or this might be what you should be saying, kind of thing. So it would be leading the participant to provide a particular response. So this would be introducing bias and compromising the validity of the research. All right. So that is leading question. And then we have under coverage or over coverage of the population. Now, what does that mean? Under coverage and over coverage are two types of coverage error that occur in your studies. So under coverage happens when members of a particular population are excluded or omitted from the sampling frame. Now what was a sampling frame? It was all the units of the population that is available for the study. Alright. So if you are omitting a particular a particular group of people from your sampling frame, that is called under coverage. While over coverage means when members of the target population are included in the sampling phase frame more than once. Okay, or when the unit that should not be included are also included. So these are the two types of coverage issues. One, where you are omitting certain people. Second, where you are including people twice, or maybe you are including people who ideally should not be included in your study. Right? So these are the ways by which sampling bias can happen in a study. Going forward, how to correct and reduce the sampling bias. Okay, so the first and foremost, you should be using what? Probability sampling, which means non-probability or non-random sampling should not be used. Convenient sampling or sampling as per own judgment should be avoided. And it should be ensured that target population is defined correctly and the sample frame is the best possible match, right? So in both these cases, you can reduce the sampling bias. Now next we have experimental and control groups. Let's see what are experimental groups and what are control groups. To start with, let's see what is an experiment. So experiment is a study that enforces a treatment or control in a controlled environment while recording the responses of the participants. So it is essentially a study that is planned and enforced in a controlled environment while the researcher is recording the responses of the participants. Okay, the independent variable is manipulated and the effect on dependent variable is recorded or studied. Okay, we know what is independent variable, right? This is the variable that is manipulated or controlled by the researcher, and dependent variable is the effect of that manipulation or control, right? So in an experiment, the researcher will be manipulating the independent variable and the effect will be recorded. And in an ideal experiment, you will have two types of groups. First would be the experimental group on which you will be performing the study, and the second would be the control group. All right. Okay, let's see what is the experimental group first. This is the group which is exposed to the independent variable or the treatment that the researcher is investigating. For example, in a drug trial, the experimental group would receive the new medication, which means if I am studying the effect of a drug on improvement in anxiety levels in individuals, I'll take two groups of people. Okay, or I'll divide my sample into two. First would be the experimental group and the other would be the control group. The experimental group, I'll provide the drug and see what is the effect. This, I will not give any drug and see the effect, right? If there is an improvement in here and the improvement is happening here also, this means it is not because of the drug, right? Because this group did not get any drug. So we use control groups to validate the findings of the experiment, right? The treatment here refers to the changes made to the variable under study, while the participants under study, which could be the human or animals, make up the group. So that is the experimental group for you, which receives the treatment or the manipulation or control of the independent variable. So now what is the control group? This group does not receive the experimental treatment, but all other variables are kept constant, which means when I'm studying these two groups for their improvement in anxiety level, I'll be putting them in exactly same environmental conditions. I'll be providing them same kind of food, same kind of what you say, entertainment, and everything else would be same. Only the drug will differ. All right. So when I keep both of these environments constant and then I'll study the result so that I can make sure that whatever changes here happening is solely because of the drug and not because of anything that they are also getting. Right. So the control group may receive no treatment, a standard treatment, or a placebo. Now, what is a placebo? It is a substance or procedure with no therapeutic effect, which means if I'm giving them the medicine, I might be giving them a sugar pill, something that mimics the medicine visually but will not have the same effect as the medicine. But they will also have that belief that they got the tablet or the drug, right? So this is the control group. A control group is formed in order to rule out outside influences or factors that may be affecting the experiment, and such experiments are termed as controlled experiments, right? So controls helps to make sure that there is no other outside influence or factors that is affecting this outcome. If there is any such factor, that factor will affect this outcome also, right? So whatever outcome is happening only here will be only because of the medicine. If it is something happening here also, then that is because of an outside factor. And such an experiment where we are using both experimental and control groups are called controlled experiments. Let's see what is the purpose of the control group. So by comparing the outcomes of the experimental group to those of the control group, researchers can isolate and measure the specific impact of the intervention or variable being tested, right? We have discussed this. Control groups helps us to determine whether the effect that is shown by the experimental group is solely because of the experimental treatment, right? This comparison is crucial for ensuring the validity of the research findings. Control group enhances the validity of the research findings, and By comparing outcomes between the control group and the treatment group, researchers can also determine if any experimental improvements are due to the active treatment or other factors such as the placebo effect, right? Then we have the types of control groups. First is the placebo controlled. So here the control group is getting a placebo, which might be a sugar pill or any treatment that mimics the actual experimental treatment but will not have the same effect, right? Secondly, we have the no treatment control, where the control group receives no intervention at all. Thirdly, we have an active control, where the control group receives an existing standard treatment used when it is unethical or to withhold any treatment, right? So this happens when you are performing trials on diseases, okay? So which means you are checking, for example, let's take an example of COVID, okay? You want to check if a particular drug is effective against COVID patients. So in these cases, your sample would be the people who have the COVID condition, right? So we'll have COVID patients and we'll be dividing them into two. One would be the experimental group and other would be the control group, right? Here, you are giving a particular drug that you are testing to the experimental group, but you cannot not give any medicine to the control group. You cannot give them a placebo, right? They are also, it's unethical. You need to treat them also. So in those cases, what you would do is you would give them the standard existing treatment, right? Which is not your drug, but the standard existing treatment which will help them also recover, right? And then you can study the recovery in phases among these two groups. So this is active control, where it is unethical for you to not give any standard treatment to the control group, right? Where you give the standard treatment, and for your experimental group, you will give your experimental treatment, right? These are the three types of control groups. Then let's see what is a placebo. So we have already discussed what is a placebo. A placebo is an inert substance, which means inactive substance or procedure designed to appear identical to the active treatment being tested but lacks any therapeutic effect. So example could be a sugar pill as in drug trials. Okay, so instead of the actual drug, you will be giving a sugar pill that mimics the experimental conditions. So this is inactive or inert substance or procedure that appears similar to the active treatment, but it is not, right? So that is placebo. We have two more classes of control group. The first is called a positive control group and the second is called a negative control group. So the positive control group, it refers to the control groups in which the conditions produce a positive outcome, right? These control groups are important to show that the experiment is being performed as per the plan, right? So it would be giving you a positive outcome. Uh, for example, maybe for example, we are studying or testing a new pimple treatment, okay? So all of us at one point or the other are hassled by the pimples on our faces, right? So there is a new treatment or ointment for the pimple that is being studied. So a positive control group would receive the cream known to be effective against the pimple, allowing researchers to confirm that the experimental setup can detect a positive effect, right? So maybe another example would be in a chemical test, a positive control would be like a substance known to produce a positive reaction with the reagents that is being used. So basically, the person would be getting something that is, that has a known act or known effect on the problem under study, right? So example, like we talked about the pimple, the positive group will be receiving something that is known to be effective against the pimple, right? But it would not be the exact same thing that is being given to the experimental group also. So the negative control group would be, they can be defined as the control groups in which conditions generate a negative result. Now these are helpful in identifying the external influences that might be existing but were not accounted for, like contaminants, right? Confounding variables that we called. Okay. So maybe if we can take the same pimple example. So the negative control group would receive no treatment at all or a placebo. Okay, so again, remember when it was a positive control group, we were giving them a different treatment for the pimple, treatment that was different from the actual experimental treatment. But here in negative control group, we will not be giving them any treatment at all, right? Or maybe a placebo to show that the baseline condition without any intervention, or show what the baseline condition is without any intervention. Like if there is no treatment given for that pimple, how it would be like, and what it would happen if you actually give a treatment. That would be the experimental group, right? So maybe we can look at it this way. In positive control, you are comparing two different treatments, both of which have an effect. While in negative control group, only one will be getting the treatment, the other will not be getting anything or might be a placebo. So this way, here you are comparing what happens when one group gets the treatment and the other group does not get the treatment. I hope you get that. In positive control, both of them get an effective treatment, but you are comparing the two effective treatments. In this negative group, you will be studying what happens if one is given the treatment and the other is not given the treatment. So that is the difference between positive control groups and negative control group. So with that, this unit is completed. As I said, this is a very small unit with just basics required for you to understand the coming units or coming chapters, right? So we'll move on to the previous year questions. There has not been many previous year question from here. So basically I could find one from December 2022. This is a part question, 515. So here you had to write short notes on the following in about 150 words each. First was sampling bias and second is experimental versus control groups, right? So yeah, that was all for this. And uh, yeah, I hope you understood all of this. Thank you all for learning with me. If you found the video helpful, make sure you let me know by hitting the like button. Make sure you share the video with your friends too. Subscribe to make sure that you don't miss out on any of the new videos. You can reach me by simply dropping a comment. See you in the next one. Till then, happy learning.